Moving to the next guest, from biology knowledge to medicine know-how. Our next guest is Loughlin, an experienced biophysicist and physicist, and now Senior Vice President Research and Early Development at Moderna Therapeutics. No need to introduction to Moderna. And of course, as a company, the Systèmes, we are extremely honored and happy to collaborate with Moderna. Moderna's aim is still to deliver on the promise of mRNA science to create a new generation of transformative medicines for patients in the areas of infectious disease, immuno-oncology, rare diseases, and autoimmune diseases. And connected really with what we just listened to with Professor Weinberg, I just noticed on June 5th, there was an announcement for mRNA related with a Merck treatment for cancer. So to tell us more about all this and this basically new generation of therapeutics with a potential that is really mind-blowing, please welcome on stage Ms. Rose Loughlin.
Thank you so much for having me. It's an honor to follow you on stage, Dr. Weinberg. So I'm here to share a little bit about a new class of medicines, mRNA. And you may have actually already received an mRNA medicine in the form of a COVID vaccine over the last few years. And I'll start by saying, Dr. Weinberg, you were right to be skeptical. So mRNA is a naturally occurring molecule. It's in all of your cells, and your body is making it constantly. And its role is really to encode different proteins that are going to have important functions for your cells and other parts of your body. And one of the biggest challenges of mRNA is that it's actually quite transient. Your body makes it, it's translated into protein, and then your body degrades it. So this is a naturally occurring molecule, and we want to take the challenge of saying, how do we turn something that your body uses into a medicine that we want to have certain properties, such as therapeutic intent. So the first thing that we have to figure out how to do is to design it. So mRNA contains 4 nucleotides, represented here with A, U, G, and C. So these are the code that tell you what protein you're going to make. So when you're thinking about designing an mRNA drug, you need to identify the right sequence. Now in groups of 3 that we call codons, depending on which order the nucleotides you put in, that codes for a specific amino acid. And so the sequence of those codons determines the sequence of the amino acids that make up the protein. And then that protein is responsible for a lot of those functions within the cell. Now, as it turns out, your body has been doing this for a very long time. And so what you can see in the blue here is that your genome has certain choices of these nucleotides and of these codons. Now, as we think about using mRNA as a medicine, we look to engineer it to be even more potent, to make more protein per mRNA that we deliver to your body. So there are a couple of considerations. So for example, these codons, these groups of 3 nucleotides, they're present in different levels and different types of cells. And if you use those that are more common or more optimal, as opposed to those that are more rare, you can actually increase the production of protein from each individual mRNA. You can also look at the structure of mRNA. So we draw it as a beautiful strand, but in truth, it has secondary and tertiary structure that are quite complex. And generally, if you move toward the more structured end, by changing the sequence, you can also have more protein produced per mRNA construct. And then finally, we don't just look at the codons in isolation. They're actually in neighborhoods. So we look, using AI, we look at what optimality we can have for codons in the context of the codons next to them. Now, to Dr. Weinberg's point, you can't just do this with computational science and bioinformatics. You have to get your hands wet. You have to generate the data. You have to generate all of these molecules and screen them so that you can actually train these types of models going forward. And what's really beautiful about this platform is that it's not just research that collects information. We make multiple medicines using this same fundamental technology. So as we work in research across programs, as we manufacture these different mRNA medicines, as we put them through clinical development, all of those learnings feed back to improving the platform so that the next medicine that we make has improved performance. And this is actually unusual. So if you think about small molecules or antibodies as therapeutics, recombinant proteins, it's actually much more challenging to take learnings from one medicine and apply them to the next. But we're able to leverage that because of our fundamental technology. So how do you turn this naturally occurring molecule into a vaccine? So as it turns out, if you want to generate a vaccine against a virus, you can look at that virus's genome and actually encode in the mRNA a protein from the virus. So when you put this in the body, your body will express that viral protein, and your immune system will recognize it as foreign and start mounting a response. So for example, your body may make lots of antibodies against that viral protein. Now, if you do happen to be exposed to that virus, in the end, your body has a head start. It already knows how to recognize it, and it has the cells that can rapidly produce more of these antibodies. Those can help prevent or diminish infection. They can also reduce the severity of disease if you do get infected in the end. Now, as it turns out, this might actually be the easy part, because if you want to develop a vaccine to impact health, you need to show that it's safe and efficacious in humans. And so, for example, during the pandemic, we needed to demonstrate that for our COVID-19 vaccine. And I don't know if you happened to try to use our healthcare infrastructure during the pandemic, but it was pretty challenging, right? Just to go visit your own physician, just to go to a healthcare facility, required enormous protocols to keep everyone safe during the pandemic, but to keep providing healthcare to people. So to be able to run a clinical trial in that environment forced innovation that had been in the works for many years, but wasn't widely adopted. It was incredibly important to be able to work with the subjects in your trial, with your sites, collect data, but as much as possible, not require those folks to necessarily come in physically. So we ran decentralized clinical trials during that time, and we still do. And of course, had a great partnership with Metadata in making this possible. The other really important benefit about taking this type of approach is that it let us look for demographics that reflected the broader population in our clinical trial. This is actually pretty challenging to do, but we think incredibly important. If you're going to show that medicine is going to be safe and efficacious for everyone, you should test it in a population that reflects your community. Now, mRNA can actually make more therapeutics than just vaccines. So if you think about the idea of making hundreds of millions of vaccines, and then start scaling down, which is also a challenge for certain technologies, but happens to be a strength of mRNA, you can think about applications in rare diseases. So a lot of rare diseases are genetic. They're mutations, just like we talked about in your DNA, but not necessarily mutations that cause cancer, but unfortunately can cause disease. So for example, if you look at propionic acidemia, in this particular disease, patients have either a missing enzyme or an enzyme that has a deficiency. It has lower activity. And so if they eat the same diet that we do and their body starts breaking that food down into proteins, they're not able to metabolize it in the same way. So with that deficiency in a protein called PCC, they actually end up with a buildup of toxic metabolites. And they can have decompensation events that can cause permanent damage and can also be life-threatening. So if you think about how you might be able to help patients with rare diseases like PA using mRNA, we actually have the ability to replace the deficient protein. So we designed for this investigational medicine actually 2 mRNAs. The PCC protein actually has 2 subunits, an A subunit and a B subunit, which form a dodecamer inside the cell. So we actually use 2 different mRNA, one encoding each of these, and the cell does the rest of the work to form that functional protein so that it has the appropriate activity. So this therapeutic is currently in a phase 1/2 trial in propionic acidemia patients. So if you go from hundreds of millions of vaccines to applications in ultra-rare populations, hundreds of patients, that's a stretch for any platform technology. And we're actually stretching it even farther in the context of oncology. So melanoma is a relatively common cancer. It has lots of mutations, as you pointed out. But within each patient, it's not that common. their actual cancer is unique. It has different mutations that are specific to that patient. Now, cancer cells can actually take some of those mutated proteins, as all your cells do, and present them on the surface. Now, the rest of your body doesn't have those mutations. So theoretically, your immune system should be able to recognize those as foreign from the cancer and destroy those cancer cells. Now, if you want to make a therapy to leverage this, you're going to have to make it individual for every single patient. You're going to have to figure out what exactly those neoantigens, those new mutated proteins are, and design a vaccine just for those to train that patient's immune system. We call that an individualized neoantigen therapy. And in order to make it, we first need to take samples. So we need to understand, for example, from a blood sample, what your healthy cells look like. Then we need to take a sample from the tumor. In many cases, this is a biopsy, and we understand what some of those mutations are in your cancer. We actually have a lot of mutations to choose from, so you need to select the ones that you want to include in the vaccine that you think will drive a vigorous immune response in that particular patient. And then, not to be underestimated, you need to make a vaccine for one person, get it to their healthcare provider, and have it administered. And so we are able to go through this cycle for patients in a matter of weeks. So if we zoom in on how we actually design these vaccines. So we look at a patient's DNA, we look at their healthy tissue, and we compare that to their tumor tissue and identify mutations. Now, we want to make sure that those proteins are actually going to be presented on the surface of that cancer cell. So we also look to see if some of those mutant proteins appear in the form of mRNA, because that would lead to translation and potentially expression of that protein. And then we also look to characterize that patient's immune system. So we can understand from the sequencing of certain HLA types how likely it is that a patient's cancer cell is going to be able to present specific mutations. And that helps us select individual neoantigens from a list that can be quite long. So we select 34 of those neoantigens and then have additional algorithms that take those little fragments and create them into one concatenarized sequence. We encode them all on a single mRNA. We design and we manufacture that, and we administer it to patients on top of checkpoint inhibitors. So right now, our individualized neoantigen therapy, mRNA 4157, is also in clinical studies in melanoma in combination with Keytruda. So you can tell that we're quite passionate about mRNA as a platform and the different applications that it can potentially have to improve human health. And we're also very passionate about global health. So in closing, I just want to share a bit about our collaborations with academic institutions around the world, where we're really focused on public health. We've actually set up a program called mRNA Access, where investigators from these institutions can use our software to design an mRNA vaccine against a neglected or an emerging infectious disease. And we will manufacture that vaccine for those folks and ship it to them so that they can use it in their own research. And so we feel like by having this accessibility to mRNA as a potential medicine, we can actually put the best minds all around the world toward improving human health. So with that, I'm happy to take questions.
Thank you very much, Rose. This is really enlightening. We have a couple of times for questions. Mm-hmm. So I have a question. mRNA, do you think it will work in conjunction with other gene therapies addressing, directing the genome? Or do you see as a replacement, not touching the genome, but really touching the expression and the translation phase?
Sure. I think you can imagine mRNA being quite complementary to lots of nucleic acid medicines. So gene therapies and gene editing, which is sort of a more futuristic technology, but is under development now as well. Certainly from a technical perspective, they're able to work on top of each other, and if designed properly, can certainly be additive or even synergistic.
Okay. Second question, after that we'll have questions from the audience. The world has been amazed by the fact that before Moderna, to have a drug on the market took a very long time. And after Moderna, nobody thought it was feasible, but you proved that with the right platform, the right tools, the right industrial processes, you could shrink. How did you do that?
How do we do it? As much as we'd love to take credit for it, I think a lot of the success there is inherent to the platform technology itself. It has a lot of efficiencies. So if you think about all of the different medicines that we make, we don't have to build a new manufacturing plant for every single one. It's the same technology, it's the same 4 nucleotides. So for us to be able to generate more medicines, different medicines, to go from very small scale to very large scale, given our platform technology, that's quite feasible. And it also happened that we were able to do this at a time when the technology, outside of our own walls, from a digital perspective, from a computational perspective, was really there to accelerate. You've seen it in the decentralized trials and the ability to capture that data and be able to analyze it incredibly quickly relative to sort of historical, more analog approaches.
Thank you for proving to the world it was feasible. Any questions from the audience? Here and after Professor Weinberg. Oh, the mic. Up the mic.
Dr. Loughlin, given the extraordinary strategy you've shown about treating melanomas, what is your prediction, your prophecy about how scalable it will be? In other words, treating one patient maybe will be brilliantly successful, but what if you need to treat 10,000 patients? What do you see in the future in terms of dealing with that complexity?
That's a fantastic question, and it is one of the biggest challenges of any individualized therapy, is when you get to the manufacturing portion, it's not computational. And again, the idea behind the platform is that we can use the same equipment, the same manufacturing, the same approaches, the same analytical techniques for every single one of those vaccines. But it is a different concept. Instead of scaling up in that world, you're scaling out.
That was a great talk, thank you. So for most drug applications, most types of composition of matter, we have to go through an IND stage and tox and all of this. So for the individualized treatments like this with FDA, how do you think about that? I mean, how are you able to get, I mean, they each have an individual composition of matter. So it sounds like innovation in terms of regulatory also. Can you comment on that?
Yes, that's a fantastic point to make, and not just in the individualized neoantigen therapy space, but also for a lot of these individualized cell therapies that have been breaking ground. The regulatory agencies are quite open to learning the new science of these technologies and understanding what their responsibility is to keep those who are brave enough to enroll in clinical trials safe. Now, safety does look different in a personalized setting, and actually, again, the concept of having a platform technology where the fundamental composition of what you're putting in is the same that you put into the COVID vaccine that we've made 1 billion doses of. It gives you quite a bit of understanding to start, and then from a therapeutic perspective, you look at that empirically.
Question here.
Thank you for your very inspiring presentation. And obviously, it's super interesting to see the way Moderna has gone selecting one modality, mRNA, to target many different diseases, either through vaccines or bringing back some proteins. My question for you is, how do you manage, we know it's not a stable molecule, and you need to reach the cells of interest. So it's one type of mRNA, but it's, if I understood well, many different types of lipid nanoparticles. And here you bring in complexity and a new type of biology. So can you comment a bit about how do you package your mRNA to target your cells of interest in the body?
Yes, absolutely. And we didn't talk about it today, but mRNA, as I mentioned, it is transient and it is fragile. So if you inject mRNA into the bloodstream, it is rapidly degraded. And so we do actually protect it in a nanoparticle made of a variety of lipids. That keeps it protected from RNases and other enzymes that would otherwise break it down. Cells can take up those lipid nanoparticles, and intracellularly, the mRNA is released so that it can translate into that protein. So we do use different lipid nanoparticles for different applications. So our vaccines share one lipid nanoparticle. Our rare diseases, which are infused intravenously, use a different lipid nanoparticle. And actually, by changing the composition of those nanoparticles, you can change their distribution throughout the body so that you can start to target different diseases in different cell types and tissues.
Thank you very much. Are we done with the questions? Oh, over there. We have tools to help you for this vectorization stuff at the nano level.
Hi. So the work on mRNA is really good in Moderna, but I had a question. How is like data integration helping Moderna in like developing medicines?
How is data integration helping us in
Yeah.
developing medicines? Yes. As I mentioned, all of our medicines do use the same fundamental platform technology. So as we advance one medicine and understand how much protein, for example, that can make in different species while we're doing research, and then we run a clinical trial and understand how much protein that makes in humans, we understand what that heterogeneity looks like. That's information that we can feed back into our own design so that the next medicines that we make build on top of that learning.
And how long does it take? What is the typical cycling time?
Typical cycling time. The slowest part of the cycling time is typically the clinical study to generate that data and get that fed back in.
Okay. If we don't have any more questions, I would like to thank again Rose Loughlin for this magnificent talk. Thank you very much. And we look forward to continue to working with you, of course. It's now time for a break. So you can catch Bob and Rose for the many questions I believe you must have. Join us in the assembly foyer and the silver ballroom, which is just outside of this ballroom for a coffee break. And be sure to visit also the 3D Experience Playground, which is just after the silver ballroom across the hall. We have many experiences for you. And we'll see you here sharp at 11:20 for our next session, where we will continue our journey on health for life. Thank you very much. See you there.